Welcome you all in the today's lecture of the plant physiology and metabolism section. We have a small topic, that is the vernalization. In this lecture, we will delve into the fascinating biological process that is known as vernalization. It is a crucial phenomenon observed in certain plant species, particularly those classified as winter annual types like Arabidopsis thaliana. This process enables plants to regulate their flowering time in response to prolonged exposure to cold temperatures. Throughout this lecture, we will explore the mechanism, ecological significance, and genetic regulation of vernalization in plants. By gaining a deeper understanding of this process, we can escalate the intricate ways in which plants have evolved to adapt to their environment and ensure their survival and reproduction. So, let's start. Just a brief introduction that vernalization refers to a biological mechanism aimed at alleviating the repression of flowering in plants through exposure to cool temperature. This process can be applied to hydrated seeds or growing plants, but it is ineffective with dry seeds as vernalization requires active metabolic processes. In the absence of vernalization, plants that depend on this process for flowering may experience delayed blooming or remain in a vegetative state. Furthermore, they are unable to respond to floral signals such as those triggered by inductive photoperiods. Often, these plants exhibit growth patterns that is characterized by rosettes with no elongation of the stem. So, moving ahead, now we will discuss about the discovery. Vernalization is a crucial process for many long day plants where exposure to low temperature is necessary to enable them to respond to conditions that trigger flowering, a phenomena referred to as vernalization. This mechanism serves as a biological clock, marking the passage of winter and ensuring that flowering occurs only when favorable spring conditions are present. The concept of vernalization was first proposed by T.D. Lysenko in 1920, who observed that subjecting winter cereals to cold treatment could induce them to behave like spring cereals. The practical implications of vernalization are significant as it allows for earlier crop harvesting, facilitates cultivation in regions where certain crops could not naturally thrive, and expedites plant breeding experiments. Typically, it is the stem apex of plants that detects the cold temperature signals, with cells actively dividing being particularly sensitive to the vernalization stimulus. The duration of cold chilling exposure necessary for vernalization varies from plant to plant, ranging from a few days to several weeks. Longer periods of exposure to low temperatures tend to promote earlier flowering. However, if the vernalization process is interrupted by heat treatment, its effectiveness diminishes, unlike the direct initiation of flowering seen with photoperiodic effects. Vernalization prepares plants for the flowering process. Studies by Melchiors and Lang in 1948 on the biennial long day plant, Hyoscyamus niger, revealed that it requires a minimum of 10 days of growth before becoming responsive to low temperature treatment. Conversely, research from the 1930s by Gregory and Purvis suggested that hydrated seeds of Petcus venteri, that is Cicale cereal, can undergo vernalization, making them sensitive to long day photoperiods. Vernalization reduces the number of long day photoperiods that required for flowering in Petcus venteri, which does not have an obligatory vernalization requirement. An experiment demonstrated that vernalization is an energy dependent process, as the supplementation of excised embryos with carbohydrates and oxygen enhanced its efficacy. Melchier's research provided compelling evidence supporting the transmission of the vernalization stimulus via graft unions, indicating a systematic mechanism of signaling within plants. He introduced the term vernalin to describe the hypothetical active agent that is responsible for triggering vernalization. Further observation revealed that once a plant undergoes vernalization, it retains a lasting memory of the cold treatment throughout its entire lifespan. This memory is perpetuated in cells derived from induced cells through mitotic division, suggesting a heritable but non-genetic mode of transmission. Interestingly, cells derived from meiotic division do not inherit this memory, indicating a specific pattern of inheritance. Lang proposed a direct connection between a vernalin and florigen, suggesting a link between the vernalization process and the subsequent initiation of flowering. These findings shed light on the intricate molecular mechanism underlying vernalization and its role in regulating plant development. Vernalization, that a process prompting flowering in certain plants after exposure to extended cold temperature, is notable in the winter annual types of Arabidopsis thaliana. As you can see here in the figure, the plant on the left side, untouched by cold, remains in a vegetative state, indicating no vernalization has occurred. Conversely, the genetically identified plant on the right, subjected to 40 days of temperature slightly above freezing during its seedling stage, has undergoes vernalization. This cold exposure triggers molecular changes leading to flowering roughly 3 weeks after the cold treatment ceased, with approximately 9 leaves on its primary stem. This scenario underscores the expected outcome of vernalization in Arabidopsis thaliana, where exposure to cold accelerates flowering, ensuring synchronization with favorable environmental conditions. Now we will discuss about vernalization results in competence to flower at the shoot apical meristem. Plants exhibit varying degrees of sensitivity to vernalization, a process crucial for their flowering regulation. Winter annuals, exemplified by certain cereal varieties, possess an early responsiveness to low temperatures, sometimes even before germination, if seeds have undergone hydration and activated their metabolic process. In contrast, biennial plants, including those that form rosettes during their initial growth phase and bloom in the subsequent season, typically requires attaining a certain size threshold before becoming receptive to vernalization cues. The effective temperature range for vernalization spans from slightly below freezing up to around 10 degrees centigrade, with the optimal range typically falling between 1 degree centigrade and 7 degrees centigrade. The impact of cold exposure intensifies with its duration until reaching a saturation point. Although vernalization generally entails several weeks of exposure to low temperature, the exact duration varies significantly across different plant species as well as the cultivars. Vernalization is the process by which cold treatment relieves the repression of flowering, is reversible when plants are exposed to conditions that counteract its effect, such as elevated temperatures. However, the duration of exposure to low temperatures correlates with the durability of vernalization effects. Vernalization primarily occurs in the shoot apical meristem, where localized cooling induces flowering irrespective of the temperature experienced by the rest of the plant. Successful vernalization has been achieved with excised shoot tips, demonstrating the shoot apex significance in the process. In case where seed vernalization is viable, fragments of embryos containing the shoot tip display sensitivity to low temperatures, suggesting the shoot apex at the primary site for vernalization. As you can see in figure, the stability of the vernalization effect in winter rye is directly proportional to the duration of the exposure to low temperature. Extended period of cold treatments results in a higher percentage of plants retaining their vernalized state when subjected to subsequent devernalizing condition. In the study, rice seeds that had been hydrated were subjected to temperatures of 5 degrees centigrade for varying durations, followed immediately by a devernalizing treatment of 3 days at 35 degree centigrade. Vernalization can involve epigenetic changes in gene expression. That vernalization relies on active metabolic process which necessitates a supply of energy in the form of sugars and oxygen during the cold treatment. Cold temperatures below freezing, which inhibit metabolic activity, are ineffective for vernalization. Additionally, vernalization requires processes such as cell division and DNA replication to occur. In some plant species, that undergoing vernalization induces a lasting alteration in the meristem capability to initiate flower development. Vernalization can trigger lasting adjustment in gene expression pattern within the meristem, persisting beyond the period of cold treatment and throughout subsequent developmental stages of the plant. Epigenetic changes denote enduring modifications in gene expression profiles that do not entail modification to the DNA sequence itself. These epigenetic modifications can be passed down to descendant cells through both mitosis and meiosis. Even after the initial cold stimulus is withdrawn, these alterations in gene expression remain stable. Such epigenetic modification in gene expressions are observed across a wide spectrum of organisms spanning from yeast to mammals and typically necessitate processes such as cell division and DNA replication akin to those involved in vernalization. Epigenetic regulation plays a pivotal role in the vernalization process in Arabidopsis. This mechanism is particularly crucial for winter annual types of Arabidopsis, which require both exposure to cold temperatures and long days to accelerate the flowering process. A key gene involved in the regulation is flowering locus C, that is FLC, which acts as a suppressor of flowering and exhibit high expression levels in non-vernalized shoot apical regions. After undergoing vernalization, the FLC undergoes epigenetic modification, resulting in its permanent silencing throughout the plant's life cycle, allowing flowering to proceed in response to long days. However, in subsequent generation, FLC is reactivated, reinstating the need for cold exposure to trigger flowering. The expression status of the FLC gene significantly influences the competence of the meristem for flowering in Arabidopsis. The FLC exerts its regulatory role by directing the gene expression inhibition of key floral signals such as FT in the leaf and transcription factor, that is SOC, and FD at the shoot apical meristem. This epigenetic regulation of FLC involves enduring changes in chromatin structure through chromatin remodeling. As you can see here in the figure, plant with a vernalization requirement typically exhibit significant delays in the flowering or fail to flower together unless they undergo an extended period of cold exposure. In cold requiring winter annual ecotypes of Arabidopsis. Vernalization leads to the suppression of the flowering locus C, that is FLC, gene expression. Conversely, in winter animals with mutation in the FLC gene, rapid flowering occurs without the need for cold treatment. A range of vernalization pathways may have been evolved. Plants requiring vernalization germinate during the fall, capitalizing on the favorable cool and moist conditions for their growth. This vernalization necessity ensures that flowering is postponed until spring, thus allowing the plants to survive the winter in a vegetative state, as flowers are particularly vulnerable to frost damage. For a plant to undergo vernalization, it must not only detect exposure to cold temperatures but also possess a mechanism to gauge the duration of this cold exposure accurately. For instance, if a plant experiences a brief cold spell early in the fall, followed by warmer temperatures later in the season, it is crucial for the plant not to mistake this short cold period for winter and the subsequent warm weather for spring. Therefore, vernalization occurs only after the plant has been exposed to a duration of cold that signifies the passage of an entire winter season. There is an there is not a universal vernalization pathway shared among all flowering plants. In Arabidopsis, the vernalization requirement is regulated by the flowering locus C gene, which encodes, which encodes the matchbox protein related to other regulatory proteins involved in floral development like delphiscence and agamas. On the other hand, cereals rely on a different protein, that is VRN2, that is vernalization 2, which is a zinc finger containing protein acting as the flowering repressor. The absence of standardized vernalization pathway suggests that major group of flowering plants initially evolved in warm climate without a mechanism to measure the duration of winter. As Earth regions gradually develop temperature climates over time, many plant groups adapted to these conditions with responses such as vernalization and bud dormancy. Consequently, these responses likely evolved independently in different plant groups in response to changing environmental conditions. So, this is all about today's lecture on vernalization. Thank you.